Today I will be talking to you about DNA replication. DNA consists of two anti-parallel helix strands which are coiled together. These helix strands consist of bases, which are adenine, thymine, guanine, and cytosine. Adenine will bond to thymine through a double hydrogen bond, while cytosine will bond to guanine through a triple hydrogen bond. The first step of DNA replication is when the two strands go through the helicase. The helicase is in charge of separating the two strands or uncoiling them by going through the middle and uncoiling them and separating the complementary base pairs. The two strands one of the strands is called 5' and the other strand is called 3'. The 5' strand is also known as the leading strand. The leading strand will, once it's separated from the 3' strand, which is called the lagging strand, will go through an enzyme called DNA polymerase number 3, which will read the template of the parental helix strand and find complementary base pairs to connect to that already existing DNA strand to form a new DNA strand, which will connect to the already pre-existing DNA strand, which will then end up with a new DNA molecule. The 3' strand goes through a slightly more complicated process. This is why it's called the lagging strand. When the 3' strand is separated from the 5' strand, it will go through an enzyme called DNA primase, which will create small segments of the DNA strand by reading its template and replicating it, which are also called Okazaki fragments. As the DNA strand continues down, it will then go through an enzyme called DNA ligase, which will bring together all of the Okazaki fragments and prepare them and form them into another backbone. It then continues its path and goes into the DNA polymerase 3, where it will bond the already existent parental strand to the newly formed DNA strand, creating another DNA molecule. And this all happens thanks to complementary base pairing. Because there is always a parental strand that connects to a new strand, which we're going to call the daughter strand, the process is then called semi-conservative. Thank you for listening.